Hey, good morning, everybody. It is time for something we have not done in a very long time. As you guys know, this is kind of a channel that has been always incorporated as part of the whole Great Debate community, which used to be about debates, isn't any longer. It's about just talking about stuff, having fun, playing games, whatever it is on the Great Debate community channel. But today we are actually going to have what's constitutive of a debate for all practical purposes. I call them discussions, but potato, potato. But it's going to be on one of my favorite subjects. Evolutionary theory. Yay! Okay, so long story short, we have Dapper Dino in here, and we have Finding Truth, Ahmed, and they're going to take actually diametrically opposite positions in here. Not this, hey, look, I don't believe you. You haven't made your burden of proof. None of that nonsense. We're going full bore. We're going two people having their own arguments, own reasoning, and their own presentations. So let me introduce you guys first. I'm going to start off for Dapper. People don't know you. Where the hell have you been? But Dapper, what up, man?
Oh, just having a nice Sunday morning. Figured I would talk about evolution, mutation, descent with modification, all that good stuff.
Oh, I got to turn to you. Okay. Wow, that was loud. Sorry,
Oh,
I'm
sorry about that.
No, not your fault. Not your fault. It's just that I have it set loud. So.
It's when you're a dinosaur, it's very easy to just blast people's eardrums off because you're very loud.
There you go. It's all normalized now. But I'd kick you down 15 freaking dB. That's a lot. Anyways, so tell people who you are and your position on this, and why we're doing this.
Yep, I'm Dapper Dino. My channel's linked in the description. The description, if I can talk. I mostly cover things like creation, evolution, but every once in a while there's a little bit of other pseudoscience in there. I've done some stuff about why homeopathy is nonsense. I do a live show every Tuesday with my co-host Bent Hovind, no relation, where we make fun of Kent Hovind, basically. Well, we don't really make fun of him. We live debunk him while I get progressively more inebriated. Except every second Tuesday of the month is actually Eric with Erica, where Gutsick Gibbon comes on, and we talk about basically the same stuff, but we give Bent Hovind a break. Thursdays, I have a scripted video that comes out every week. And then every once in a while on Saturday, I will do another stream or video. I'm currently working on a Leaving Young Earth Creationist video for hopefully not this Saturday, but the next one maybe. Although maybe this Saturday. So yeah, that's me.
Awesome.
I am, of course, taking the affirmative on the question, is descent with modification through mutation and natural selection true?
Awesome. I like that. I like when people take a position. All right. Anyways, we also got Finding Truth in here, who I don't know very well, but has been a member of the Great Debate community on Facebook. Seems to be very polite. Never had an issue with him. Don't know his positions very much, but we'll give it a go. So, Ahmed, what do you got to say for yourself and what you do? What are you going to argue with here?
All right, so I run the Finding Truth channel, and I'm an engineer, an electronics and communications engineer by education. I also did my master's courses related to computer science and computer engineering. I have a master's in business, and I have 4 years education in Islamic theology, 30 years experience in computer science field. So I work with design all the time. I design systems and implement them. I've been an executive for 20 years in transnational corporations. And I think that evolutionary theory suggests that all what we see around us comes through randomness and natural selection. A random process is a blind process, and natural selection is blind to its objective. There is no objective function. It contradicts with everything that I have been taught in engineering. And I just find it not logical at all. It is one of the subjects I've been fascinated with since my teens. So I've been looking into this for like 30 years. And eventually I thought I will put up a channel, and I will discuss religion, and I will discuss also science, and I will see where they reconcile and where they don't. I feel that there is a great deal of systemic bias in the field of biology, and that the matter of evolutionary thinking is extending not only in the matter of origin of species, but also in the origin of life or abiogenesis, even in the origin of the whole universe. And I think this position is more or less spawned by philosophical naturalism and manifesting itself in a methodological naturalism aspect in science, which is okay, yet invoking something like randomness answers no questions whatsoever. And my position is that this is false. It is flat out false. The one thing that I don't like about it is the random part, that random mutations are what is driving innovation, and that natural selection can fix that by filtering the right mutations in and filtering the bad ones out. My argument is that this is not possible, and I'm going to give plenty of examples about that.
Awesome.
On the theological side, I think that, so I'm a Muslim, so I think that in general, whether you are following an Abrahamic religion, a Jew, a Christian, or a Muslim, or other religions that believe that there is God who wants to be found, I think that God who wants to be found is not, it does not make sense, given that this is the position of religion, that God obscures himself to the extent that when we look at him, when we try to find him, as far as how life was created, we find randomness. I think this does not make sense. It doesn't add up to the position of religion about a God that wants to be found and worshipped.
Awesome. All right. Well, yeah, that was a pretty good intro. So you're a smart dude. We're going to just give you that. Okay. So Dapper, you have a lot. You have a challenge here, man. So, make the evolutionists proud, because you got to get that NASA shill bucks here. So we're going to start you off first. You're going to be the one doing your first presentation. I'm going to give you around 15 minutes, give or take, timed. But of course, it's not going to be inflexible. So go ahead and start your presentation, and let's see what you got.
All right. Well, thank you very much for having me here, Steve. Ahmed, thank you for coming. I appreciate it. It's always fun to have a chance to have a hopefully polite and civil discussion on these topics. So I actually want to start out talking about the things that I am not here to debate, because I always think that it's important to get out of the way what the debate isn't about, so that we can focus on what it is about. So one of the things it's not about is the origin of the universe. Evolution, descent with modification is compatible with a large number of possibilities for that, including many of them involving gods and miracles or anything like that. We're also not here really to talk about the origin of life, because the origin of life, the first life form, obviously can't have descended via mutation and natural selection from an earlier life form, because it was the first one. So we're also not here to talk about that, because the process of mutation natural selection doesn't explain abiogenesis. We would have to look to other areas for that. But since this is about modification through mutation natural selection, no abiogenesis. In fact, while I do plan on addressing things like general common descent, even that is not technically part of the discussion necessarily. So for instance, if it turned out that all birds were created separately from all mammals, but that all mammals shared common ancestry, that would in fact confirm descent with modification through mutation and natural selection. So it's not even strictly necessary to argue for universal common ancestry, although I may end up touching on those areas. So I just wanted to set that up as very important things. I also don't really intend to discuss theological implications of this, other than to say, if what mainstream science says is true, then it does preclude a few, but not all, theological positions. So if you find that troubling for your theological position, I'm not going to try to modify mainstream science in order to make it fit your theology. I think the onus is on you to modify your theology to make it fit with reality. So that is why I do not plan on addressing theology very much. So then we come to the actual topic, descent with modification through mutation and natural selection. So first we're going to go in order. So we have descent. So this is something that is obvious to everyone. It has been for thousands of years, probably tens of thousands of years. Organisms descend from earlier organisms who are their parents, or in the case of single cell organisms, the one cell splits into two daughter cells, and you can think of the original cell as the parent, and the two new daughter cells are the descendants. So I don't think there is any debate that descent happens. Now the next part is modification, and we're going to take modification and mutation together. So while many mutations will not actually cause any change to the phenotype or the actual way the organism exists in terms of its size and shape and interaction with the environment and chemical processes that go on within it. Some of them will. We know this. We know this because we've seen mutations in the wild and in the lab cause phenotypic differences. So then the next question is, okay, well, do mutations occur during the process of descent? And the answer is also obviously yes. We can measure it happening. We can take a look at over the generations. We can watch mutations accumulate in the genomes of organisms. We've done it with humans. We've done it with many model organisms, lots of bacteria, all these things. So we know that descent with modification through mutation happens. And then the last question is, does natural selection filter out some of these mutations? Well, we know this is the case for a number of reasons. One of the best ones is simply statistics. So for instance, you can take a look at what's called a pedigree mutation rate, which is simply the mutations that are accumulated from parent to child. But if you then, sorry about that, if you then go further generations, so you take parent to child to grandchild, and then go to great-grandchild, and so on and so forth, the rate of these accumulating mutations start to go down. And one of the reasons for this is that if these mutations are detrimental, then they will be unlikely to be passed down. And so over generations, that mutation rate is lower to what's called the phylogenetic replacement rate. And the biggest factor in that is going to be natural selection. But we can also watch natural selection happen. One of the most famous examples of natural selection happening, which is in the peppered moths in England. So we know that the melanistic morph of the peppered moth, which made them black, didn't really exist before about 1820. We can tell this both with genetic clocks in terms of just actually taking genetic samples and looking back to how far back the mutation is likely to go, but also because we don't get historical records of them appearing from naturalists until around the 1820s. So we have two independent methods of saying this was a novel mutation. And then we have the observation that the ratios of melanistic to, I suppose, original morph moths flipped radically before the Clean Air Act was established in Great Britain, which cleaned up the soot from the trees, essentially. And then when the soot started clearing up from the trees, the ratio flipped back. So we know we had descent with modification because we have a novel mutation. And we know that natural selection caused the general distribution of the different morphs of these moths to change. So we have the whole thing. There is really no question as to whether or not natural selection causes changes in an organism through filtration of these negative alleles. But we also have other examples. For instance, new mice populations in the last few decades have started to enter into the sandy beaches around Florida. And while the original populations tended to all be black or brown, there is a rare agouti morph which ends up with them having a much lighter color. And in that area in Florida, that has been heavily selected for. And now light sandy colored mice predominate because they are better at blending in with the sand dunes. But we also have other examples where we can trace back mutations around things like ring species. Now, there is no perfect ring species in the world, but we have things that come very close. And so we know that essentially, if you go to adjacent populations, they can interbreed, and we can trace back the mutations causing it. But when you get to the two ends of the ring, they tend not to be able to breed. And so this is going to be a result of at least descent with modification through mutation. Now, we also have things like, and I know Ahmed is aware of this, we have the Lenski long-term experiment with E. coli. And in that, not only do we have descent with modification through natural selection, granted the environment itself is artificial, but the selection is not. Humans are not specifically picking out bacteria to propagate. But in this, not only do we have the morphological evidence of the changes in time, but we also, because samples are taken of each population, we actually have essentially like an actual historical record of the genome that can be revived to check on the actual genome, their performance, and things like that, which is how we found out that the citrate, we found out that there were two important mutations for citrate metabolism in the populations in an experiment that can undergo aerobic citrate metabolism, which is in fact a defining characteristic of E. coli, that they can't do that. We also know of other instances in bacteria, such as the production of nylonase. We know that nylonase wasn't really produced until after the substance nylon, and some time after the substance nylon was produced. And we found out that there were bacteria eating nylon in essentially nylon factory wastewater. And so, again, we have mutation that is selected for through natural selection. We can also observe this in antibiotic resistance. And if we want to go to the fossil record to see modification, we have extremely good fossil records for many organisms, including both Centrosaurian and Chasmosaurian Neoceratopsians. We have an excellent fossil record for Paravian Maniraptoran dinosaurs. We have an excellent fossil record for the human line, at least from the point of Australopithecus going forward. Before that, it gets a little bit spottier, but that's still a very smooth transition there. We have to the point of almost seeing every single generation. If you look at the fossils going up through the vertical extent of the strata in diatomaceous earth, where we're getting all these diatoms, or if you're looking at the evolution of coccolithophores in limestone, you can see almost every single generation represented. So we know that descent with modification is happening. There really is no debate about that. And we know that it can occur through mutation, and we know that natural selection must filter these things, because it is simply a mathematical inevitability that if organism A in the same population as organism B produces more offspring than B, overall, those genes will come to dominate, or those alleles will come to dominate the genetics of the population over time if that situation persists, that A is consistently more successful than B because of natural selection. So I'm not really sure what argument can be put forth to say that descent with modification doesn't happen, or that it is not affected by both mutation and natural selection. I see it as it's as close to mathematical certainty as one could get without actually being in mathematics proper. All of the premises we know are true. Descent happens, modification happens, mutation happens, natural selection happens. Unless you want to deny one of them, I don't know how we can say that the conclusion that this statement here is false. And really, I could go on with more and more examples. We know divergence time for things like Arctic hares versus the hares that live in other parts of North America. We know that they generally can't breed if they're far enough apart in their range. We can trace back the genetics. We can even trace back paleogenomics in some cases all the way back into the Triassic to look at things like the mutations involved in taking the sort of basal tetrapod jaw configuration and turning it into the weird mammal jaw configuration. So even then, we actually have a good idea of exactly which mutations occurred in which genes that are conserved across vertebrates. And so I'm honestly kind of confused as to why someone would decide to take the negative in this. But I think with that, I'm just going to end it because I'm interested to hear Ahmed's points and actually get into the discussion.
There we go. Yeah. All right. You actually had a little more time than you had, about 5 more minutes, but I guess you're going to relinquish the balance of your time, which is fine. So you had about 10 minutes. Ahmed, you get the full amount, though. He's giving you the advantage here, man. He thinks you need like a handicap, so he's giving you 10 minutes to your 15. So make it to your advantage here, man. Show him up. All right. It's all yours, man.
So thank you, Dapper, for the introduction, and it was quite good comments. I took some remarks. I'm going to respond to your comments after I do a brief presentation. So I'm gonna share my screen now, and we have some fun with the presentation. Okay, so is it on now, Steve? Can you see it? Hello? Yeah, okay. So here I already introduced myself, so we can skip that. This is the address of my channel, so we can skip that. And let's first start by saying what's okay for me. So I have no problem whatsoever with all established science that is causal, testable, and observable. This includes genetics. This includes inheritance, Mendelian inheritance, with all the recombination, everything. This includes the recently discovered epigenetics within the couple of decades ago. This includes breeding, genetic drift, natural and artificial selection, all sorts of adaptive responses. And in terms of mutation, I believe there is mutation, but they are predominantly deleterious. And maybe sometimes they can be neutral, but the observation, the evidence says that mutations are deleterious, and we are going to go through that in more detail. So now my background is engineering, and in engineering, we use science and art to serve human beings. So engineers are those guys who use science to create real things that work. And the centerpiece of engineering is information. And engineering something starts by having an intent and having a purpose for that thing. And when you have a purpose for a thing, then you have a design for that thing that serves the purpose. And the design reflects the information that is coming from your purpose. And then you manifest that design into a set of instructions to build that thing. And when it is like a machine, then there are systems, and these systems have parts, and these parts are built to work together. And then you make operating instructions for these things to work. So if you're building a car, the car has the information that is the design of the car, but there is also the information that is, how are you going to use the car? And this information goes from the designer to the operator. So for you to operate the car, you need to receive an information from the designer, from the original equipment manufacturer, to be able to operate the car. Now, if you are creating a fully autonomous car, which is something that I think will be coming soon, you will need to give the operator less information. So we'll go into the car and tell the car, take me to Dallas. And the car then has to have a set of instructions so it is able to operate itself. And this information is the superset of information that the operator would have received. And when we look around us, we see those creatures that are manifesting these things. We see that there is general purpose for each creature. We see things like bacteria that are cleaning up everywhere. We see scavengers. We see plants that are feeding animals. We see a very big integrated system with subsystems of living things living in it. And we also see the extra set of information, which is quite amazing. So we don't just see a creature that moves, but we see a creature that has extra information about where it is going to. We see amazing things like birds that don't only know how to fly, but they can also use the magnetic field of the Earth to make something like a GPS positioning for itself. And it knows that if it is going to migrate, it should migrate from this place to that place in that season. So it has this extra information about how to operate itself in the bigger thing. And this cannot make any sense unless everything is designed. And the designer is not only delivering information in the genetic material that is manifesting itself into a creature that is living, but also downloads information to these creatures so that they can operate themselves. And so we're looking not only at cars, we're looking at autonomous cars, we're looking at much more than this. We're looking at cyborgs all around us, animal cyborgs. And then we're looking at humans. And this is it's an information-centric thing that we see around us. And with our knowledge about what it takes to have this working, we need a processing system, we need programs, we need a database, and we need operating instructions, and we need integrated systems. And these things do not occur through any random process. There has never been an engineer, and there will not be an engineer, who will design something that works through randomness. We see that today, as engineers, for very sophisticated problems, we use algorithms like genetic algorithms. We use algorithms that are very heavy on trial and error, but we give these algorithms an objective function that represents the objective and the purpose of the designer. And those objective functions see where the design should go. So natural selection, which is totally blind, which has no sense whatsoever of what is the objective of any series of mutations, will have no idea about which mutations are really to be picked. So natural mutation selection cannot fix the randomness of mutations. I have just, from my background as a communications and electronics engineer, looking around me in nature, just this morning, jotting down what we are learning from living things. And some of these inventions we have created long ago, and then we get struck that they are already existing in living things, and we modify our designs so that they work. The very late nanotechnology, we get shocked when we look at the molecular biology level, and we see those nanomachines inside of us. And none of those inventions has come to us through a random thing. They have come through people who have purpose and intent and put together designs. And the existence of these things that are designed into living beings only show, if we look at how things are in operation, that those things are designed. They are not the product of any random process. I'm putting here things like radar, sonar, GPS, firmwares, information coding and decoding, error corrections, hard drives, neural networks. Even neural networks, when we're thinking of a very sophisticated calculation mechanism, we look into how our synapses and neurons are working, and we imitate that so that we can get out with artificial intelligence. We have CCDs, fiber communications, cameras, bigger things like planes and submarines, microphones, robots, nanotechnology. All of this are designs that are there already. Can this or can this not be explained by genetic material that gets random mutations, and those random mutations will then survive one way or the other, and then one mutation after one mutation? You have mutually constructive mutations. My position is that this is totally false, it's totally impossible, and it's totally absurd. I can, go into discussions about mathematics, etc., but I would rather, as an engineer, take a different approach. Because as an engineer, if you tell me this thing will work this way, I will tell you, okay, show me. Show me. If it works this way, show me. So let's have an engineering approach to this. Let's have a hypothesis, and let's advance the hypothesis and see if randomness, which is random mutation, combined with blindness, which is natural selection, can give us anything. Yes, natural selection would naturally, because something that gets a beneficial mutation would reproduce more, it will have a better chance for survival. But we're not talking about the chance for survival here. We're talking about one mutation that makes somebody will see the light, and then another mutation that makes him see black and white, and then another mutation that makes him see colors. And we're not talking about one mutation, we're talking about thousands upon thousands of mutations. So if there is no system that sees an objective function that has a direction, this is completely hopeless. But let's look at this. I'm going to show here something. I'm not sure if the audience know some of these things, but let's look at the latest of the latest of our technology, the future, the hope of humanity, nanotechnology. And there are these things today that we want to design to put into our bloodstreams or whatever, nanobots. And when we looked into molecular biology, these things are already there. I'll just show these glimpses, 1 minute each almost. And they're just amazing. I will let them speak for themselves. So just a second.
Just to let you know, you're about 5 minutes, give or take, okay?
Yeah. Thank you.
I know that Ahmed had earlier said that the video clips took about 5 minutes, so if he goes a little bit over just showing the video clips, I don't mind.
All right, thank you.
All right.
Thank you for that.
While you're doing that, since you're looking for something, let me give acknowledgement to Rez Instance for the $50 super sticker. Thank you, Rez. Really appreciate it, man. Really appreciate it. Glad you're enjoying this. Let's do it. Go on.
So can you guys see the screen now?
I can see that it's buffering.
What we're seeing now on the screen is this incredible thing. This is what is called a kinesin. And this is a molecule, a protein molecule that is moving inside each and every cell of our body. This thing that is moving on two legs is a protein, and it's moving on what's called a microtubule, and it's moving material around. And this is the kind of thing that we would dream of. This is a single molecule. Can this be a product of a random mutation? Now let's see what else this thing can do to just to appreciate what these things are. So here's another thing. And what we're looking at now is what this thing does. It's buffering a little now. And the guy that we're going to see is the guy who has actually discovered those things. And those things are all over the place in our bodies, and they do pretty amazing things because they're moving things around whenever we need them to move things around, and without them we wouldn't survive. So this is Ron Valley, and he's talking about this. I think you cannot hear the voice, but this is how this molecule moves. It performs these reactions, ATP reactions, and it does 4 reactions, and each reaction will make it lift a leg and put another leg. And here's this thing moving small beads. This is very microscopic. And the great thing is that this thing, which is the equivalent of a truck in the cell, there's billions upon billions of them inside of us. And they're moving things, and they're moving it at 4 times the efficiency of the best car that we have ever built. And if you consider the size, they would be even moving much more faster. And here's another example of what these things can do from that other video. And I would really recommend that people would go to these videos and see them in full, because I personally, I found them completely amazing. And yeah, I'm not sure if it's showing this. I don't think it's showing it. Let's see this one to appreciate the beauty of this. Let's see this thing in operation, in making us who we are in the cell division. And what this does, this is something called the kinetochore, and it is this process. These are the fine tubules that we have seen before, and these are these tubules are stretching the chromatids, the chromosomes, to split them. And there's this red signal that says, I'm not ready yet. And you can see how these microtubules are building themselves up. And then you can see these amazing machines starting to move up the microtubules, carrying a signal. And then the signal, when the chromosome material is ready to split, the signal changes. And those kinesins, and there will see other motors moving around, dyneins, are gonna carry the signal, physically carry the signal, carry the stop signal away, and tell the cell that I'm ready to split. And then the splitting happens. And the first time I saw this, it brought tears to my eyes. This is super duper, super crazy intelligent nanotechnology. And these guys, the dyneins moving and the kinesins moving, the long legs so that nobody, this is completely mind-blowing. This is hyper sophistication. How does this thing occur in the cell? How can the first cell, first eukaryote at least, have this kind of sophistication? Where did the information come from? How does something this sophisticated happen? When this thing fails, cell division fails, and you do not get an embryo, there is no life. There is no replication, there is no life. Life is complicated at its very basic level. It's not that, that life was very simple and it became very sophisticated and there is a gradient. Life starts from an extremely sophisticated point. It does not start simple, stupid, and become super smart. And the last thing I want to show here is how this thing looks in terms of, I cannot show it for some reason. Those tubules, just for your information, they are not just stretched there all the time. They form together something called the cytoskeleton, which gives the cell its shape. But also, when some material wants to be moved from one place of the cell to another place, those microtubules self-assemble. That was the fourth one. And they just stretch like magically in front of the protein motor, so that the protein motor can walk, carrying the proteins or whatever it's moving around from this point A to point B, and then the microtubule dissolves. Imagine this: you want to go from New York to Brooklyn, and then a bridge just happens to appear in front of your car, and then a car appears that you ride, and then when you're there, the whole bridge dissolves. This is like science fiction. And now, if somebody tells me this is random events taking place, please tell me which random event has taken place first. Is it the random event that has created hundreds upon thousands of genetic material to create the proteins first, the kinesins or the dyneins? Or is it the random events that created the microtubules, the actin fibers, or whatever other materials? There are thousands of these motors in our bodies. And when they were traced back, they were found to be ubiquitous. Life depends on them. How do these things happen? I'm going to go very fast, 10 seconds each. If random mutation is correct, and in the beginning I said I'm fine with many things, I'm not fine with random mutation. Life is not based on randomness. We're 150 years away from Darwin. We are like 60 years away from neo-Darwinism, and we expect that if randomness is driving this, then number one, the major transitions of life should be explainable by random mutations and natural selection. They are not. If you start at the edge of abiogenesis, which is having a protocell, the transition from a protocell, which should be simple, stupid, to the least universal common ancestor, Luca, is not known. The transition from prokaryotes, which is, maybe Luca was like a prokaryote, but prokaryotes are very sophisticated. Bacteria is very sophisticated. Yet the transition from prokaryotes to eukaryotes is not explainable by random mutation and natural selection. Absurd theories like endosymbiosis have to be invoked. The transition from eukaryotes, which are unicellular in nature, to multicellular organisms is not explained by random mutations and natural selection, and other absurd theories like colonial theories have to be invoked. So number one, there's no explanation for every single major transition in the history of life is not explainable by random mutation. You can put on top of that, how did sexual reproduction come into place? How did marsupials happen? How did placental things happen? Nothing is explained. The major things are not explained. Number two, we expect to have a smooth gradient. We don't have a smooth gradient in the history of life, because if you are invoking small random mutations, very, very small changes, then we should have very smooth gradient for life. We don't have that. We have things like the Cambrian explosion. We were all told billions of years, 3.5 billion years, 4.5 billion years. But all life as we know it today, every single big animal that we know today, or marine creature, occurred only 540 million years ago, and through a period of somewhere from 7 to 15 million years, everything that we know today has happened. Does this make sense if the main driver is random mutation? Absolutely not. We expect also innumerable transitions of life forms. We don't have any transitions of life forms. Yes, we have many fossils, but I have asked questions to evolutionary biologists, to people who are very entrenched in this. I just ask a very simple question. Start from a cow, a modern cow, start from a modern horse, and trace back the exact ancestral lineage of that horse or cow or rabbit or fox or any other terrestrial mammal, and tell me which are the specific, don't tell me the class and it was this kind and this kind and that kind of a family. Tell me this is the animal, this is the predecessor animal, this is the specific predecessor, and show me the fossil. There's nothing.
Okay, I need to start wrapping. I need a conclusion coming up. By the way, man, I've given you 10 more minutes of time. So, what's your summation, real quick? Give you 1 more minute to wrap it up.
So,
1 minute timed.
All right. So, yeah, I don't need to go more like. So we have the genetic mechanisms. We have genetic code that is against mutation. We have mutations that are muted when they happen. We have an abundance of examples. We expect abundant examples of beneficial mutations, but we only see deleterious ones. So how come mutations are the driving force? We expect that plenty of junk DNA. ENCODE project showed that we are like 60 to 80%. In bacteria and simple organisms, junk DNA is almost nil, just 2 or 3%. Non-coding DNA turns out to be coding for things beneficially all the time. We expect things to be not efficient, but we are learning efficiency from nature. We expect that there are no jumps, but when we look at the human condition, we find language that is unmatched by anything else. We find intellect and we find free will. All of these things are great jumps in the history of life. And if mutations are what is driving this, then I can say comfortably, then nothing makes sense. All what mutation gives is a state of laziness, where when we know something, we don't know something, then we say it's random. This is not the proper position of human intellect. We should get out of this randomness claim and look into what's really happening if we want science to advance.
All right, that's a good summation there. All right, so you guys, okay, so I'm going to have you guys back and forth now. Let's be clear here. I've listened very carefully to both of you guys' presentations. You guys came from a very, very different perspective. Dapper's talking about natural selection. You're talking more about intelligent design. I see the disconnect already, but I want to see you guys have a back and forth on this. I don't care whether you start from Dapper's side, dealing with the peppered moth, Coccolithus spores, talking about natural selection, or if Dapper wants to dive right into your things with the Keynesian molecules and transportations and how meiosis and all this stuff. Take your pick where you want us to jump in, Dapper, because he had plenty of time. You can either go really from where you're starting to, because there is a huge disconnect between you guys' presentations. But I'm going to turn it over to you guys. I have a free-form back and forth, maybe for about 20 minutes to 30 minutes to see how it goes. And then we'll take some questions. I do have one question from the audience real quick. I'm not going to have you guys answer it right now, but I'm putting it on the table for later so you don't forget, if you don't mind. And Danish Debater had asked for 50 DKK for the Q&A, so you guys don't forget. You can prepare for it now. From each, if you had to select one of each outside of religious works, thank you. Can you mention one book and one paper to support your views? But we'll get back to that, and you'll answer that during the Q&A, but I don't want to forget about it. So in the back of your mind, have some paper or book that you want to recommend. But Dapper, it's all yours.
All right. So there were a whole bunch of things brought up. Unfortunately, I think most of them fall into what I call the gee whiz category, which is basically, look, this thing in some organism or lots of organisms is really complex. Therefore, but that's not an argument. If you want to say that something is complex, therefore mutation and selection and drift and the other mechanisms of evolution cannot produce it, you have to actually produce reasons for that. So all of the things about showing really complicated biochemistry, not in itself an argument. And I was actually kind of surprised that, Ahmed, that you brought up genetic algorithms, because the thing is, while it is true that the selection criteria were set up by humans, it is in fact random changes to computer code that are in fact themselves producing complicated engineered plans, at least, not actually the physical objects, but then humans go and make the physical objects and they do work. And so what we have in life is simply, rather than having an artificially produced selection criteria, we have the same thing, except it is simply, did whatever the organism is successfully manage to keep reproducing and reproduce better than its conspecifics say. And so just the fact that we can use genetic algorithms is itself very high confidence demonstration that random mutations with non-random selection can in fact produce very complicated things that in some cases humans can't understand. There have been instances of humans using genetic algorithms to create machines that they don't entirely understand why it works. We've actually seen this with genetic algorithms producing airplanes that are aerodynamically stable, but that no human designer would think would be. They just end up being so.
Dapper,
go
do
ahead.
you want me to jump in here and we close point by point, or how would you like to go through with this?
Yeah, if you want, I'm open to you responding to those few things that I just said there.
Because I'm taking notes, but I think they will accumulate. So if you'd like, we can just close them one by one. So you can bring up something, I respond to it.
Sure,
the way
go ahead.
that you want. So you mentioned two things. So the first thing is that I say these things are too complex and they cannot occur by random mutation. Now, yes, now we need to look at a very specific point here. Now, what is the default logical thing? Is the default logical thing that a complex system is a design system, or a complex system is the result of randomness? So I would claim, why I'm showing this is that I would claim that normal human reasoning is that complex things are products of design, of a mind. And if we find that life has started at very, very simple starting point, and then we can see very, very small additions to that, we might think of it. But the issue is, these things that I have shown and other things that are even much more complicated have been there since the beginning. Actually, we don't know what was there before them. So as far as we can observe, life has started from a very complicated point. So if somebody is claiming it started with a gradual thing, there is no gradual thing. So this is why I'm bringing that. The second thing is that, yes, I did bring out genetic algorithms, and it would be peculiar for somebody who is arguing against randomness to bring a genetic algorithm, but I'm a person who likes to confront things. And in my field, in computer science and in communications engineering, communications problems are very sophisticated. We cannot solve them. We need to use the genetic algorithms. And what genetic algorithms essentially do is that they change features of a circuit, or for an antenna, or for whatever, aerodynamics of a plane, so that we can find a solution. But how do they work? You give the genetic algorithms exactly what you want to do. It will not generate a plane for you with new aerodynamics without knowing what you want to do. It will not generate a new antenna in my field without knowing what you want to do. You have to give it what is the gain that you have to give it, the beam width that you want. You have to give it this and that, and you have to tell it these are the components that you can change. And to make it reach a solution, you give it ranges for these changes. And you put an algorithm for selecting the outcome. Otherwise, it will never converge. So if somebody says, we have this thing that is generating errors, mutations are errors, they are copying errors during reproduction, or they are defects caused by radiation or whatever, we have errors. They are errors, they are not trials, they are errors. Genetic algorithms use trials. Mutations are errors. And natural selection is blind. There is no objective function. It doesn't know what you want to design. So those things are nothing like genetic algorithm. So this is my reply to you. Why I brought it is because I want to confront this to the end. I have gone into numerous discussions with people who are even trying to emulate genetics and biology using computer models. And when we look at the facts, guys, these are not random things. You're putting an objective function and you are moving towards it. And with this complexity that we're seeing, this is nothing like random mutations. Genetic algorithms are nothing like that.
All right, so there are a couple problems there. One, like I said, there is in fact a fitness function that is objective in the world, which is simply comparative reproductive success, as well as simply not dying out. And we know that there are objective things that are done for this. At the very basic level, life forms are just organisms that maintain a thermodynamic disequilibrium while using energy gradients, whether they be chemical, thermal, photons or whatever, in order to power their processes that keep them at thermodynamic disequilibrium with the environment. So when that ceases, that is a selection against. And when they fail to reproduce at a higher rate than their competitors, that is also a weaker form of selection against. So we know we have observed instances of random mutations with selection criteria, both in nature, in the case of many observed instances of evolution of novel features of speciation in both the lab and in the wild. And we also have examples of random mutations with selection in the case of genetic algorithms. The fact that a human has set up different survival criteria than nature doesn't change the fact that given a selection criterion, which we have in nature, and random mutations, in both cases, there is no reason to think that we cannot get complicated and to some extent hard to understand systems because we observe it happening today. And in fact, one of the things that you pointed out is we were going back a little bit to abiogenesis. I don't particularly care to go back to abiogenesis because if you wanted to have that topic, it would be a different debate. But you did also mention that how we go from prokaryotes to eukaryotes is apparently some mysterious thing. But the thing is, we have observed endosymbiosis taking place, which is essentially the difference between prokaryotes and eukaryotes. Is eukaryotes are a lineage which has taken in other organisms as symbionts, things that live with them inside the cell. And we can see this now. For instance, there's a kind of, well, I'm going to call it a protist for ease, even though I know that the word protist is no longer really scientifically accurate, but people know what I mean, called Pollinella. Now, most species of Pollinella are predators of cyanobacteria. They go and engulf them, take them apart chemically, take all those nutrients, and produce waste. But a subsection of them actually have started taking cyanobacteria and then use them directly as a new form of chloroplast. And in some of these species, we're actually seeing some of the genes for photosynthesis transferring from the bacterium member of this new organism, what is now the new chloroplast, and being transmitted into the nuclear DNA of the Pollinella. And we also have observed instances of organisms going from single cellular to colonies to actual true multicellularity. For instance, we've seen it in various lab lines of Saccharomyces cerevisiae, which is brewer's yeast. It's how you get, beer, basically. And so given certain selection pressures, such as filter feeding cellular predators, or some other experiments have involved centrifuges that separate out, larger organisms from smaller ones. Other ones have done settling experiments. The things that settle to the bottom of the test tube or flask or whatever are taken out. And in these cases, we've seen Saccharomyces cerevisiae, which is a single-celled organism, go to clumps of cells, and then from there start to actually have divisions of actual function in the cell, which is the definition of multicellularity. A multicellular organism is a collection of cells where not all the cells do the same thing. And so the very basic version of that is you have somatic cells and reproductive cells. So the somatic cells are involved in all the metabolism, doing producing the structure of the organism and whatnot. And then the reproductive cells are just there to create new descendants of that organism.
Real quick, Dapper, I want to make sure we get Ahmed in here a little bit, so there's a little more back and forth.
I was starting to ramble.
Yeah, that's okay.
Yeah.
That's what I'm here for. This is what I do.
Okay, so let me respond to a few things of what you've said, which is you've raised really good points, but I think each of these points might as well be a debate on its own. So let's first look at the matter of natural selection will pick things based on survival, and then you mentioned endosymbiosis, and then you mentioned transition to multicellularity, which are things that I mentioned in my presentation. So I will take this reverse order. So let's look at multicellularity. So you mentioned the filter feeding thing, and I think you're referring to the experiment, maybe the one about the paramecium that is acting as a predator versus algae. And let's look at what happened in this experiment. Essentially, algae is a kind of creature, a unicellular creature that has many modes of existence. They exist in single cells and exist in colonies. And there are many algae and many unicellular organisms that will go in colonies when the conditions call for it to cooperate. And there is no multicellularity in this experiment. What happens in the experiment is that the algae that were living alone under threat, they colonized, and they maintained the colonization even when they reproduce. And in some of those experiments, even when the predator was removed, the colony mood of existence remained for many generations. However, colonies are not multicellular organisms. Colonies are colonies. And there is no cell differentiation. There is no specialization that occurred.
But there was in Saccharomyces cerevisiae.
I have actually a long video on that, or a few videos on that, and maybe I will need to refer to that specific experiment. But people would like typically to invoke things like Volvox, and they say that look at this algae that is a transition state between unicellularity and multicellularity. It's a colony that acts as if it's an organism, and it does not. And all what's happening is that the algae that are on the rim are using their flagella to move, while the algae that are inside use their flagella to split the cells apart. And if one of the algae on the rim wants to act as one that reproduces, it just moves its cilia inside, and it just does the thing. And if you separate one of them from the inside or the outside, it will just do the whole thing. They will just survive, and they will form new colonies.
So
you're not
we need a clarification.
Yeah.
What is it that at a minimum would make a group of cells multicellular as opposed to a colony? Because I do agree that there is a difference, but I want to know what it is for you that makes it cross that threshold.
That's a very good question.
A collection of cells.
That's an extremely good question, and this question has a philosophical aspect, and the philosophical aspect is, what is life? So if you look at, so here's my hand, and my hand has many cells in it.
Yep.
But the identity of the creature is me. I am the organism, and those cells are part of me. The one that wants to survive is myself. So the cells in my hand, they receive an instruction to die, and they die. And when they do not die, they become a disease. They're called cancer. So programmed cell death
can get cancer?
Yeah. I'm sorry?
So I really want as concise a definition as you can. Is your definition multicellular organisms can have cancer and colonies can't have cancer? No?
So
okay, what is the difference?
So the definition, the borderline, in my opinion, is whether the identity of the organism is held at the multicellular level or at the individual cell level. So does the individual cell strive for surviving or not? Or is it that the whole set of cells strive to survive? This is number one. And number two, that for them to be an organism, so some of the cells will manifest the DNA in a completely different morphology. Some will look like a kidney cell, and some will look like a skin cell, and some will look like a neuron. And those cells do not look at all like each other because they have completely different functions. They will go into that morphology, and they will stay into that morphology.
Okay, so can I summarize that?
Yeah, we'll make them stay like this.
Let me try to summarize that. So we have cell differentiation and apoptosis. If a group of cells...
Cell differentiation and identity.
Well, I don't know what identity means.
Identity is at which level does the organism strive to survive? Is the organism striving to survive at the multicellular level, as this is one unit, or is each individual cell wants to survive? And I can give you a little example if you would like, but
well, we're at the hour mark, and I think we've kind of diverged from the actual topic a little bit. I want to kind of try to bring it back a little bit, but not to shut you guys down on this for a little. So a little bit. I have some own questions as well. I got 2 super chats. I'm gonna get them out of the way. I want to wrap this up real quick about your dialogue when it comes to multicellularity and like some kind of colonial type group. For example, like the Portuguese man of war. Would you consider that to be a multicellular animal or colonial type animal? And what do you mean by identity? Because I was kind of lost on that too, what you meant by identity. Because even for a group, when they have a species that wants to somehow survive, so even as a multicellular animal as opposed to a colonial group, the goal is survival. And the whole topic is that survival based on natural selection. Is there going to be a descent with modification from selective pressures that this animal or whatever unit is going to survive through mutations? I think that's the intent of the topic. So real quick, wrap it up what you mean again by the difference between multicellularity and some kind of colonial group. And then I'm going to read the 2 super chats and then kind of hopefully get it back on track. Okay, sound good?
Yeah.
That works for me.
All right. So you want to wrap it up real quick, Ahmed?
Yeah. So for me,
the
transition between a colony and a single or multicellular organism is 2 things. One, colonies can incorporate relatively unrelated organisms. So not all the cells have a single genetic ancestry. Multicellular organisms, they do all have a single ancestry, whether it be simply from, an embryo that developed asexually from a parent, or whether it be, an embryo that is the combination of 2 other organisms. And also that there be cell differentiation, which is morphological. So if you can divide into somatic and reproductive cells, and the whole group of cells has a single common genetic heritage from a single original cell or group of identical cells, then we can call it a multicellular organism. We have seen the development of such organisms multiple times in different disparate taxa, both in the lab, and I'm not sure if we have any observed instances in the wild, but we do know that it can in fact happen, and we know that it happens through mutations because we sample the genetics and we sequence them, and we can keep track of which genetic changes happen. And it turns out that actually there aren't actually that many genetic changes that you need to go through to go from single-celled free living to a colony, and then on to having differentiation and having gametes that go on to produce new groups of cells that are themselves a multicellular organism.
Okay.
That is my wrap up.
Sounds good. All right, so Macabre for $10 straight said, that's not a question, but he says, ancient con man mooches off old women, rapes children, and commits wars of crimes. It inspires modern engineer to argue that science and bad. I feel bad for humanity. Stupid Whore Energy for $5 says, what does Ahmed think about examples of endosymbiosis like Carcinella rudi, a sequenced genome that is on the path of the symbiont and organelle? If you're familiar with that work.
Okay, let me first answer your question here about the multicellularity. Do a small wrap up. So,
sure.
To my knowledge, there is no experiment where you have started from a single cellular organism and you ended up with a multicellular organism, which means that it has cells that has the same DNA that has gone into perpetual specialization, and then it can reproduce and form a new creature. And one of the reasons
you haven't looked for them, they're numerous and they are trivially easy to find. So that you aren't aware of them, to me says that you haven't looked for them, and that might be because no one has brought them to your attention. But now that someone has, I'm hoping that you will look into them, because it is a well-known type of experiment that can be repeated many times, and each time you do in fact result in multicellularity, often through different pathways, which means we have multiple ways to get there. So it's like telling me no one has ever bred cows. It's just not the case. And that you've never heard of it doesn't really change that.
So I have actually heard of it, and I have actually looked into specifically the papers that claim that, the one about the yeast, the one about it's called the de novo origins of multicellular life observed in the lab. And I have read all those papers. I have them. I have my remarks on them. And when you read the exact paper, you will find that this is just a multicellular colony. There is no organism there. But we can have that on a separate one. But the one that I want to point out here is that one of the reasons that life is so difficult to move from single cellular to multicellular organism is that when you create a colony, there are those guys who are called cheaters. And what a cheater is a unicellular organism that contributes to the colony, but it takes more than it gives. And guess what? The more the colony lives, the more cheaters happen. And the more cheaters happen, natural selection favors the cheaters because they're taking more and they are more efficient on energy. And the other cells know nothing about what's happening. And eventually what happens is that those cheaters cause the colonies to break down. And this problem is an unsolved problem. and it happens over and over again. So
It's
when we
actually
look,
not
All
a
right.
problem.
So let's get back to the
Yeah.
the thing here. So Supernova Energy has that question on the table. What do you think about the examples of endosymbiosis, like Carcinella rudi and the sequence genome between the path and symbiote of organelle? Because you had mentioned something earlier that for some reason you had denied endosymbiosis, even though the mainstream, and I'm sure Dapper would agree, the mainstream approach is that there has been some form of endosymbiosis through the mitochondrial, the matrix, right? That's why we have extra ATP production for animals. And then plants have had something similar happen to them. But you've denied that. But what do you think about the examples that Carcinella rudi has had for the sequence genome that he did?
okay, so let me take the mitochondria as an example, because this is a very important, and even the cyanobacteria, which is claimed to be the precursor of the photosynthesis in plants. But let's look at the mitochondria. So you have the mitochondria, which looks like another unicellular organism, another prokaryote called rickettsia, and it is claimed that it is essentially mitochondria is a rickettsia-like endosymbiont, and at one point of time it went into another cell, and here we go. And you look at the rickettsia, you find that it has a few thousand genes. You look at a mitochondria, you find it has 37 genes. And there is a rule in evolutionary biology that homoplasy is not evidence for homology unless you establish ancestry independently. So if mitochondria, in some of its features, like for example, it has ring-shaped DNA, looks like rickettsia, which is a prokaryote that has ring-shaped DNA, this is not evidence on its own. And we go back to what is life. When I have a bacteria in my gut, and we have plenty of bacteria in our guts, are those bacteria still independent organisms, or are they part of me? The consensus is they are independent organisms. They want to survive regardless I survive or not. If they are into symbiosis with me, they are good. If they are not, they are disease. But when you look at the mitochondria only has 37 genes. Some of them are code for RNA, and just 13 of them, if I remember correctly, code for proteins. And those proteins are the exact proteins that are needed for the enzyme complexes that are involved into the energy production thing. So the mitochondria is essentially a powerhouse for the cell.
Right.
It has nothing else.
But the question
So
is, I want to bring back to the question a bit, because I think you're
Yeah, so the answer to the question is, if we are claiming that this is endosymbiosis, then we are redefining symbiosis, that one of the symbionts is sacrificing its life. This is not what endosymbiosis or any symbiotic relation is. Symbiotic relation is both live and both are making benefit. So I really find that this is a misnomer, that invoking symbiosis to explain eukaryotes is one of those, mental gymnastics that evolutionary theory pulls off every now and then, when the normal order of things, which is a smooth gradient, fails to be fulfilled by random mutation and natural selection. It happens all over the evolutionary path, in all of those 4 points that I have mentioned. One of them is the formation of eukaryotes. Whether now we are going back to cyanobacteria, which is forms the chloroplasts, claim to form the chloroplasts, or to rickettsia, which is claimed to be a rickettsia-like thing that form the mitochondria, or any other thing. You will go into the organelle, you will not find a full DNA sequence, or a very, very small thing that is very specific to its function. It is not a living creature. And why would a creature go there and sacrifice his life? And if there are things that we see into other endosymbionts and any other symbiosis relation, show me the path. At what point of time did the endosymbiont decide to give up its life? And
but real quick, hang on one second. Her question is, though, on the C. ruddy, which is the supposedly one of the smallest genomes out there of a symbiotic relationship, okay? She's asking, though, how did it go from a symbiont to the organelle itself? Because it was engulfed, right? And being an obligated, it was an endosymbiotic relationship. At some point, there were two individual things. They came together, right? And now it became more from a symbiotic relationship to an organelle, right? And the same thing would be with the mitochondrial DNA. That is separate DNA, right? It is an organelle in the body. She's asking, how did that come about if it wasn't through like natural selection? Through because the whole topic of discussion here is how do these things come about through descent through modification, if not through natural selection? Now, Dapper, you're arguing that it's from selective pressures. You're arguing that there's no real teleological aspect to it. That's not to somehow a function. Because I remember you came in here originally, Ahmed, when you talked about function, right? So I think Sarah's question, Super Horror Energy's question, is really on point here to the whole discussion itself of why do we have things like endosymbiosis? You could deny the mechanism, but why is it that we have those things that work through some kind of seemingly natural processes for the overall benefit to form something like an organelle for the animal?
Yeah. So the first step in endosymbiosis is, of course, the larger organism engulfing the smaller one and then failing to actually kill it, which can happen for a whole host of reasons. I don't think we really need to get into every single one of them since we can watch it happen. And so at this point, now you have an organism which is going to become an organelle. Now, if it is going to survive, it will have to reproduce with the host. And any host symbiont relationship in which the symbiont is providing benefits to the host is more likely to have the host evolve to support it and vice versa. Now, you can say, why would it get rid of some of its genes? Or why, in fact, it generally doesn't get rid of a lot of genes. What actually ends up happening a lot of the time is the genes will transfer to the host organism's genome. And then certain genes that might be shared between them, the copy on one may end up being deleted through a deletion mutation or something like that. But that the gene is still going to exist because if it's necessary, it will persist in either the host or the symbiont. And so really, all you need to know for why would the symbiont choose, it doesn't choose. It's now in a situation where the options around are mutations that will make it more likely to reproduce with the host, which will be selected for, and mutations which make it less likely to reproduce with the host, which will make it selected against. There really isn't any big mystery about this. Everything is exactly as we would expect. And the reason that endosymbionts tend to have small genome sizes is exactly like I said. They tend to only carry a few genes that are actually vital to both the symbiont and the host. And those are the only genes that have any strong preservation on them. So, things like deletions or mutations that break genes that are not necessary for the symbiont don't get selected against. And that really is it. In fact, like I was saying with Pollinella, we can actually see instances where the cyanobacterial genome is being, in some cases, transplanted into the host Pollinella nuclear genome. And so some of what in the original cyanobacteria would have been its own genes for producing various proteins involved in its photosynthetic functions are now actually being produced by the nuclear genome. And they're actually transplanted from them. We can tell which area of the original cyanobacteria lost this gene and where it was inserted into the Pollinella genome. And this is a thing that's currently happening. We are watching a new kind of blue-green algae evolve before our eyes via endosymbiosis with known still extant groups, in both cases, both the symbiont and the host, that are free living. And we also have instances where they are, in fact, in a organelle and host situation. So we're just watching it happen right now. We don't even have to worry about whether or not it could happen in the past because it is currently happening. It's like someone telling you that trains can't work while riding a train.
So the question there is, Dapper, at one point, does the endosymbiont decide that it's better for it to die? Because we're not looking at one or two genes here that have moved. We're looking at more than 95% of its genome lost.
And
when you lose 95% of your genome, you have died. Your hand is not a separate organism.
Well, my hand has the same gene as the rest of me.
Yeah, exactly. So now this organism that has a different identity, that has a different life of its own. Why, from its own perspective of natural selection, natural selection is all about survival, yes? It will undergo changes if this helps its survival. Mm-hmm. So number one, where is the survival if you lose 98% and you become somebody's generator? This is not survival. This is you have died. Become a part of something else. And then,
are they still around?
yeah,
That's
you
the
see where I'm
selection.
coming from.
No, I don't.
And then
what you're saying makes zero sense to me.
And then what changes? What is the process where you would change from ring-shaped DNA into the normal double helix DNA that the eukaryote has? Where is this process, and where did the nucleus of the eukaryote come from anyway? Because both cells were supposed to be prokaryotes, and prokaryotes do not have nuclear membranes. And where did these perforations on the nucleus come from? And how did the nucleus of the eukaryote let in?
We're shifting goalposts. We're not going to shift the goalposts. The question right now that you proposed before you started shifting the goalpost was, how is it beneficial to lose large amounts of your genome as an organelle? Then you started asking new goalpost shifting questions. So I'm going to ignore the goalpost shifting because that's a dishonest tactic, and it might not be intentional, but it is what happened. Now, to answer your original question about losing your genetic information or losing large sections of your genome, like I had said, those parts of the genome in an endosymbiont, which are no longer required, no longer have positive selection in order for them to be preserved. Therefore, they tend not to be. It's the same reason that when you see cave fish who have been living in caves for long generations, because the genes regulating things like eye structure and eye development are no longer positively selected for, they tend to be lost. And the reason that you can say that that is selected for is simply because, I mean, look at any extant multicellular eukaryote. They're full of billions to trillions of mitochondria. Mitochondria have not gone extinct. They're doing quite well. In fact, very few eukaryotes have ever lost them. There is one possible group of single-cell eukaryotes that I think it's not clear if they lost them or they're actually basal to when eukaryotes first got mitochondria. But we know of an animal with no mitochondria. It's in fact a myxozoan, which is a kind of cnidarian, and they are parasitic cnidarians. And one species of these doesn't even have mitochondria. But overall, eukaryotes maintain multiple mitochondria per cell. So mitochondria are doing just fine in terms of survival. And that's all that's needed. Natural selection doesn't really care about your personal assessment of whether it's better to be a free-living bacteria with a large genome or whether your personal assessment is that it's better to be a tiny organelle with a small genome. As far as natural selection is concerned, your human biases towards one of those things doesn't matter. The fact is, after being engulfed by a larger cell and not being digested, it is beneficial for both organisms to produce mutations which allow the eventual creation of the current situation where we have the mitochondria with a tiny genome essentially playing powerhouse for larger eukaryotic cells.
So genome size doesn't matter?
No. Yeah. It only matters if the genes in question are actually important to survival. But once they no longer are,
they're no longer required.
Selection ceases to operate on them, and they will go away over time.
So as long as the mitochondrial DNA is producing the extra ATP, because normally we'd only get ATP from like some kind of glycolysis process or something, right? So because animals use a lot of energy, right? Because we have that extra ATP production now, allowed for multicellularity, it allowed for more complex structures. And I think Ahmed would probably argue that that isn't something that could be described normally, but from some kind of just random stochastic natural process, if I understand his argument correctly.
Well, there's selection, which is not random.
Let me comment here about the moving the goalposts, Dapper, because there is no moving of the goalposts. What I'm saying here is, this is what is claimed, that you have a creature that has like 5,000 genes, and it goes down to 37.
Okay.
So for it to go down to 37, the claimed thing is that the 4,900 and something moved from its own genome and went into the genome of the host eukaryote, right or not? So
not in all cases. Some of them did. Some of them almost certainly did. But some of them probably simply were lost over time through deletions because there was no selection pressure for them to stay.
So let's look at the status quo. The status quo is what makes the mitochondria itself is not the 12 or 13 genes that produce the proteins that are needed for the enzymes for the energy production. The body of the mitochondria is manufactured through proteins that are expressed from the nuclear DNA in the eukaryote host. So what I am saying is, for this process to be claimed that it is a result of an endosymbiotic reaction, then we need to understand, if there is no mechanism, then it didn't happen, because this is science. If you cannot prove it, it didn't happen. It's not a hypothesis, right or not? So we have a creature that has 5,000 genes. It becomes 37, and miraculously, those 5,000 genes were building the whole creature. Now, the other genes that build the rest of the creature, the rest of the proteins and whatever, the lipids and whatever else this creature needs, are expressed from the nucleus. So how did this migration happen? For this migration to happen, ring DNA has to be converted into double helix. It needs to go through the nucleus, which is a very protected, the nucleus has a cell and a nuclear wall that is very protective, that had pores, that will only allow things in that should come in. You cannot just inject DNA into the nucleus of a cell if it doesn't want it. So there is no path. The issue is that there is no path. Yes, you can claim that there is endosymbiosis because mitochondria looks like rickettsia, or because chloroplasts look like cyanobacteria. But they can also claim that it's the same designer who is designing everything. So now, the thing is, if this is an endosymbiotic reaction, then show me the path. How will 4,000 and something genes move into the nucleus? This is now the question. Now, this question is not solved. We can claim that it happened, but unless we can solve it, it remains a hypothesis. And
so I'm sorry,
the thing is,
horizontal gene transfer doesn't happen?
Horizontal gene transfer between what? Between ring DNA and double helix DNA?
Yeah.
You are looking at a eukaryote. You are not looking at two bacteria who are exchanging genes of the same type.
Right, Ahmed, we know of current historical instances of eukaryotes transferring known genes from known bacteria that they have engulfed into their actual nuclear genome. This isn't an unobserved thing. We know of it happening currently right now. You are in a world where this is currently happening. This process that you were saying can't happen is currently happening. Like I said, your entire argument about endosymbiosis has been like someone who is currently riding a train and claiming that trains are impossible for various reasons, mostly incredulity. Your incredulity does not negate the fact that it is currently, right now, in 2021, a thing that is happening. Why is it that you would persist in saying this thing that is happening right now can't happen?
Because I believe that we are not witnessing it happening. If you have a paper that shows that it is happening right now, that ring DNA from a creature that is an endosymbiont to another one is being transferred to the other one to the extent that it's losing 98% of its genome and becoming an organelle, okay, let's look at the research. And if it's happening, it's happening.
Say we go back to, we're 10% into this losing of the 98% of the genome. Would you tell me, after presenting you with a paper like that, well, because it's only, it's only lost 14% of its genome, this doesn't count? Would you say that to me? Because I suspect that you would.
Well, now again, if you gave a good example here. So you have a fish or a creature that's living in the dark, and it would like, and it doesn't care really much about vision.
I would like that question answered though, Ahmed, real quick. Could you answer that question?
Yeah. So if a creature loses a part of its DNA that it doesn't need, it's okay. But
Chief, could I get that question answered?
If it loses everything except for the need of another creature, it is not okay. This is what I'm saying.
Okay, I would like you to answer the question that I asked, though.
All right, what's formulate the question specifically, Dapper?
So the question is, if I show you a paper about a currently ongoing process of endosymbiosis, but the genome of the endosymbiont has not been reduced to the point of 98% yet, but it is in the process of that, will you come back to me and say, well, it hasn't lost enough of the genome, therefore this doesn't count? Because I suspect you might.
I would look into the paper for this part of its genome that it's lost. What functions does it correspond to? If those functions are essential for its being, for its composition, and it has lost it happily to the host, then I would accept it.
Okay, but what if it's only done 10% of that?
It's not a matter of the percentage, Dapper.
Okay, good.
It's
good.
It's something was without. Okay, so real quick.
It's the matter if it's losing part of its genome that will make it unlivable, unsurvivable outside of the host.
Well,
Okay.
later on,
Okay.
either today or in the next few days, I will send you some papers on endosymbiosis in Pollinella, and I look forward to your response to that.
All right, so let me ask you guys this. So go on. Take for example the cavefish, right? Everyone knows the story of the cavefish. You know, they live in darkness. They don't need their eyes. So any genes relating to eye function, it could be lost through whatever deletions or whatever. It doesn't matter because it's not energy efficient to the fish. The fish rather use that energy for something else. So it doesn't really care about the eyes. We all think everybody on that page. So the question really is, and going back to the title of the discussion. What makes that happen, right? I mean, if natural selection is simply, and I honestly think a peppered moth just could have been really done. Honestly, I think that kind of was the example, the definitive example of natural selection. But
I agree.
But if through somehow these selective pressures, it is, hey, look, this fish no longer needs these genes. Whether the genes are deleted, whether they're just non-functional, whether it's junk DNA, makes no difference. The fact remains that those genes are not going to be around because they're just wasted energy. And that is to the benefit of the fish, the organism itself, for survival through natural selection. Now, Ahmed, you say, since you're taking the other approach, if it's not through natural selection, what is causing those genes to no longer be important to that fish because of the conditions that the fish is in, such that it doesn't need a size? That to me really is the crux of this particular discussion. Would you guys both agree on something like that?
Yeah, I think that's fair.
All right, so go ahead, Ahmed.
I have no problem that natural selection exists. So if this is a specific creature that's living in the dark, there is genetic entropy taking place all the time, and there are deleterious mutations taking place all the time. So the ones that see and the ones that do not see, they become disabled due to a mutation, will have the same chance to reproduce. And since seeing is not an essential function to life when you are in the dark.
Can you tell us what you mean by genetic entropy? Because I know the term, right? I know the term from Stanford's. What was the name? Stanford.
I think it's Stanford.
Yeah, but that is not in any literature I've ever seen as far as science. So, when you say like genetic entropy, what do you mean by that?
So So let's say this. So the genetic material of any creature is degrading with time because it is accumulating mutations. We can agree on this, right?
That's not true, no. I 100% disagree that there have been numerous experiments attempting to observe this kind of thing, which is actually in science called error catastrophe. And error catastrophe is nearly impossible to induce. In fact, I know someone who tried for quite a while to induce error catastrophe in a population of bacteriophages. And while he did eventually get the populations to extinct, even then he still couldn't actually show that it was error catastrophe. And quite frequently, despite saturating all of these bacteriophages with as many mutations as mathematically could possibly happen, they would persist and entirely recover in the course of him going home for the night and then coming back in the morning to check on his cultures. So no, genetic entropy is a completely nonsense idea. It does not happen. There is nothing to it. It is bunk.
So back again to what I am saying. So if you are accumulating errors, the errors that will make you not care about it, so you get an error that causes your eyes to be blind and you are living in the dark, so you will have the same chance to reproduce. But if you get an error that will make you also deaf, then this error will be selected against, and you will just, the other ones that are not deaf are going to survive. So I'm essentially saying the same that you are saying, that there is the aspect of survival that will balance out these continuous mutations that are happening. So if we agree on both those things, number one, we agree that mutations are deleterious in nature. So where do the innovations come from?
Well, we don't agree that
well, hang on. I think this. Hang on. Let's stop there for a second because I think because you just kind of threw that out there. Everybody agrees. Hang on, Ahmed. So let's talk about what you just said. Just be fair, because you threw that out there, and I think there's obviously going to be pushback on there because, correct me if I'm wrong, most mutations are going to be more in the neutral category. There are deleterious ones, but the beneficial ones are the ones that are going to be selected for over a period of time. That's the whole point of complexity. That's the whole point of descent with modification, which is the topic. So even if there's going to be deleterious load of some kind, there's going to be some kind of deleterious errors that are going to be cropping in. Those are expected to be, I guess, weeded out, phased out, because the organism is less likely to survive. Meaning that the ones with the more beneficial mutation, rather than the deleterious ones, are going to be selected for if there's not a neutral mutation, right? So if it's some mutation that has a phenotypical characteristic that can be acted upon, it is more likely the species or the animal or the unit itself that has the beneficial mutation is more likely to reproduce. And that's the whole thing behind it, is reproductive success, right? And so I did throw that out there about the deleterious things. I don't think Dapper would agree with that, right, Dapper?
I would not agree that mutations are on the whole deleterious. I would say that most mutations, because you got to remember, most mutations end up in no phenotypic change because either they're in non-coding regions, or they result in synonymous codons, or even they can change a protein, but the change is not actually going to affect how that protein functions. Because you got to remember, most proteins are actually fairly robust and can undergo a significant amount of change without actually having any measurable.
You can have an amino acid
on
substitution and still have a function of okay.
So then, after we set aside neutral mutation, it is the case that in any given fitness landscape, most actual random changes to phenotype, which are going to result from mutation, will take you away from a fitness peak. But because you are in fact on a fitness landscape, some of the random directions in which the phenotype can move are in fact to higher fitness peaks. And the only way that this could not be the case is if you're already literally on the fitness peak, in which case any phenotypic variation will end up being selected against. But generally speaking, because fitness landscapes shift, organisms are almost never managed to get literally to a fitness peak in their local fitness landscape, and therefore there are almost always available beneficial new phenotypes, which means that there must be, by definition, available beneficial mutations. And because any mutation that in principle can happen is likely to happen at some point in many populations, for instance, humans are mutation saturated. There are so many humans that every mutation that could occur in a human genome is likely already to have occurred somewhere to someone. Therefore, there will always be beneficial mutations that can be selected for.
So when you have such a high amount of iterations, right? So many people have in their genes. You're arguing that it's likely that certain people are going to have the same exact mutation.
Yeah, actually, we know. So I guess I can't say for certain, but if you work out the math as to how many mutations could possibly happen and how many humans there have been over the past few thousand years, it is almost certainly the case that every possible mutation that could happen to the human genome has already occurred. Mm-hmm. And since we know that some of them have to be beneficial, we know that there have been beneficial mutations in humans. And we actually know of a few in particular. For instance, lactase persistence, which is actually an increase in the strength of the promotion of the lactase enzyme production. So what happens in lactase production is, normally in most mammals, you have a promoter for lactase, and it ends up getting swamped by other signals. And so lactase is not produced at a sufficient quantity to continue to digest lactose. But in many humans who have lactase persistence, which I suspect actually Ahmed is in that group because I believe he lives in a region where that's common, where people can eat dairy. The promoter region is actually strengthened. It promotes lactase more. And so later and later into life, you continue to make enough. Although in some cases, you don't make quite enough, which is where you get to mild forms of lactose intolerance. So you get people who can eat a little bit of cheese here and there, but if they sit down to have a bowl of ice cream, that's going to cause problems.
But that's a good example. The lactase persistence, that mutation actually has occurred more than once in the population. I believe one time it happened, and then 10,000 years later, or something like that, they occurred again and was selected for, if I'm not mistaken, on the lactase persistence. But
that may be untrue.
So I'll pick an example. Let's go with lactase persistence, Ahmed. Since the topic is ascent with modification through mutation and natural selection is true, and you obviously would agree that lactase persistence would be a good thing, right? Normally, as you said, the promoter causes it to shut off at a certain age. I don't know, like 2 years or something. So we're no longer able to produce the enzyme to break down lactose at a certain age, and that's why the persistence it's so good. It gives us an alternative nutrient source, right? That has been selected for through natural selection, but you're saying that is false, right? That's the whole thing from the topic of discussion. It's false. If it's not true that is being selected for from natural selection, what are you positing that's causing that particular beneficial mutation? Because you have a whole new carbon source here, a whole new thing to get energy from, which is beneficial. What are you causing that is causing that selection? What are you positing as causing that selection?
Okay, so actually, Dapper and yourself have raised here 4 good points. So let's take them in sequence. So number one, the people who argue for genetic entropy do not say that catastrophes happen. They argue that mutations that are typically more or less neutral or very, very slightly deleterious take place. And since their effect is very slight, they do not get selected against. But their accumulation will eventually cause an issue to the whole population after some time. So this is their claim. Now, whether this is what is happening or not needs to be tested. However, we see that if we acknowledge that DNA is not predominantly junk, which occurs to be discovered more and more every day, that what we're thinking is junk is not junk, then changes to functional DNA will typically cause damage.
What are you using the term junk for?
What do you mean by junk? That's an open question, because not everyone has the same definition.
Exactly, yeah. I've noticed that as well. So when you're using the word junk, do you mean non-functional, non-coding, no conformational changes? When you say the word junk DNA, what specifically do you mean by that?
So what I see is that one of the predictions, or one of the part of the worldview of the mutation paradigm, is that genome gets extended by continuous transpositions and duplications here and there.
What do you mean by junk?
And that those duplicates have no function.
Okay, yeah, right. We understand. But what do we?
We have 98%. There is a perception that the human genome has 98% doing nothing.
But what do you mean? Okay, but this is what we're trying to. I don't mean to cut you off, but that's what we're trying to ask you. When you say non-function, do you mean non-protein coding, or there's no conformational change? There's no detectable activity, because when they did the project on that, right? They basically redefined a lot of these things, and they said, "Hey, look, if we detect anything, even a spurious transcript or a conformational change, we're going to call that somehow a non. We're going to call that some kind of function to it." The term junk DNA, typically, if I'm not mistaken, refer to non-protein encoding genes.
Yes, which is not to say that it doesn't do anything.
It could have mRNA. It can have other things that it does, but it just doesn't produce a protein. Well, is that how you're using junk DNA, or?
If it produces RNA, if it regulates something, whatever, then it shouldn't be called junk. However, the initial perception was that only a very small part of the human genome did something, and the rest was junk. And then with time, we understand that both introns and extrons have function, and that, there was this ENCODE project.
Yeah, the ENCODE project, yeah, exactly.
At one phase of the project,
they claimed
they had a very low bar on what would consider to be functional, right? So if it did have any regulatory functions, it would consider to be a functional thing, right?
There are some problems with their definition. One is that anything like spurious transcription, where a bit of DNA ends up as being transcribed into RNA, and then that RNA just does nothing. They would call that function. Also, they called methylation targets function, even though methylation at a particular site in the genome actually tends to restrict the ability of that site in the genome to actually function. So far from methylation targets in what I would say is being evidence of activity, it's evidence of inactivity. In fact,
I agree.
Viruses are often methylation targets to prevent them from producing diseases. So the ENCODE project's definition, even by the admission now of some of the members of it, is broad to the point that it is useless. Using their numbers to tell you how much of the genome is actually important for the phenotype of an organism, in this case humans, is not very useful.
Yeah, and that's why I like the difference between junk and garbage DNA, Dapper.
So for instance, if something is functional because it's a methylation target, and then it mutates, there's not likely to be any phenotypic change, because the fact that it was a methylation target in the genome means it likely wasn't being expressed anyway. Well,
Ahmed had when he said earlier, I think he said, because he was talking that he accepted things like, I think he said evo-devo, but he accepted epigenetic as well, right? I mean, Ahmed, I think that you mentioned that, right? So you accept that methylation things happen to exist. There's epigenetic factors that could be selected for. They're probably not a long-term thing, but they can actually be going from one generation to another, but probably not for very long persistence. But those things you do accept, right?
Of course, but
okay.
Let me complete my argument here, because so
sure.
we're looking at existing function for the lactose. We're looking at part of the genome that we see stopping this function for some humans and not stopping it for other humans. And it happens that those humans happen to be guys who take care of cattle in Africa and some parts of Europe. And both of those populations developed lactose persistence after at a specific point in history where they became breeders. Now, this takes me back to an experiment that Dapper here has mentioned, which is the Lenski experiment, which is a very indicative experiment, because what Lenski did is that he put those bacteria and he deprived them, so they only have citrate around. And the initial impression was that a mutation has taken place. But if you really look at what has happened, you will find that the bacteria, the E. coli in this case, had all the machinery to metabolize citrate. And that the only component that changed was the transport part to enable the citrate to infiltrate the cell in a proper way. And that what happened is that the cell took a reaction where a specific part of its genome was copied to the active part, which used to transport glucose, and now this part can transport citrate too. So I'm very skeptical when somebody calls this a mutation, because it's not. It is a part of the genome that has been moved under a specific stress condition, and it didn't happen randomly, because it even happened in 2 groups out of 5, if I remember correctly. And since this experiment claims that it is a repeatable experiment, it means that anybody who would do it other than Lenski should get the same result. So it means, and even I think he used the term acceptation for that. So I would claim that many things that we as humans, with our very limited knowledge, start by calling something junk and then take notice that it's not junk, start by calling something a mutation and then notice that it is an acceptation, or that it is an adaptive response, or it is an epigenetic response. We only know about epigenetics 2 decades ago or 3 decades ago. So I think that invoking randomness as the solution for everything that we do not understand, that we find that a single nucleotide, a single base pair has changed in a part that we used to call an uncoding region, it causes the lactose intolerance to stop. And it causes that very conveniently in the populations that are breeding. And how did this spread? Some say that this happened because, you know, a kind of famine happened, and then people who could drink milk survived. And then we have to make up all those stories, because we have to fall back to a random event. While the more we learn, we find that epigenetics is doing things, that the cell have these amazing mechanisms. We've just discovered CRISPR. We know that our genetics are so sophisticated. We know that we have error correcting mechanisms and gene editing mechanisms that are totally unimaginable if we go back a few decades ago, just 10 or 15 years ago. We knew nothing about these things. So if you look at the reality of the progress of our understanding to what happens in our cells, everything, in my opinion, the more we see that it is not randomness that is driving this thing, the more we see that this program, this information repository, this DNA, the genome, and what is on top of it, the epigenetics thing, and who knows where epigenetics come from? Where does this information come from anyway? And what are the limits? What are the limits of epigenetics? Can epigenetics be, for example, 5 or 6 or 10 years from now, be discovered that it can re-edit the genome itself? Not only does the methylation and not only does the histone wrapping, maybe it can do something else. So why?
If it edits the genome, it's not epigenetic, it's genetic. That's the definition.
The epigenetic is beyond the genome, right? So if it has an effect on the genome, it can't be epigenetic.
It could be an environmental cause for mutation, but that is not epigenetic. That's what.
Do you think there's a real quick? Do you think there's a fundamental? Oh, go ahead, finish up.
What if there are other mechanisms like CRISPR that are editing genes upon need? What if this thing that happened in the Lenski experiment, where a part of, in my point of view, this has been an intentional rewriting. This is not a random transposition that happened in the Lenski experiment.
You think it's intentional? You think that's an intentional thing? There's intentionality. Okay. So let me clarify for the audience here.
Reach down into Lenski's lab.
Hang on.
Population of bacteria that is starving, and there is only citrate, and suddenly citrate transport is activated. How on earth can we think that this is a random
Well,
mutation? It is not.
let's look real quick. We are running back in time. I'm going to put you on the hot seat real quick, Dapper, okay? Because he's been on the hot seat a little bit. And I think it's fair that you are. But let's, for the audience, recognize that natural selection doesn't claim to be a stochastic process. Natural selection isn't random. You can have variation that's random, and then you can have acting upon that. But natural selection itself, nobody, I think, claims that that is a random process. But Dapper, so he makes a good point. I mean, I knew the answer to it, but I wanted to put you on the hot seat. So he's talking about Lenski's experiment. Now, the metabolic pathway already existed in the E. coli. It just wasn't for an anaerobic condition for the citrate metabolism, right?
Yep.
Because the bacteria in 2 of the flask, I believe it was, I think you mentioned, which is right. This occurred. This out of 20,000 whatever cycles of iterations of generations, this mutation occurred more than once. And he's saying that if we do the exact same experiment, shouldn't we expect that at some one of the flask we should have this exact same mutation? Is that what you're asking, Ahmed?
It already occurred in 2 flasks, not one.
Yes, 2 flasks. I agree. So we should be able to repeat that experiment and have that happen at some point, correct?
Yes.
Okay.
It is possible for it to happen again, but there are a couple of things to point out. One is the 2 populations in which it had happened actually shared an ancestor population in which a priming mutation had occurred. And without this priming mutation, the subsequent mutation would not actually allow citrate metabolism.
All right, but what would happen if we had the original population to do the experiment with?
If we reran the experiment and we had a population size that was big enough to saturate mutations, such that every mutation that can occur is almost certainly occurring, then because both the priming mutation and the subsequent mutation that actually allowed for citrate transport into the cell are both possible mutations, we would expect that it is indeed possible for both of those things to happen again. The question is, why is that not random? If I take the entire set of all random possibilities, and it includes these 2 possibilities, and then those 2 possibilities happen in a small subset of my population, that is entirely consistent with random mutations causing this citrate transport to become an option for these E. coli. And the fact that it didn't happen in all of them, I would say, argues strongly against the idea that it's an intentional thing. If it were intentional, if the bacteria in some way or some higher power were intending for the bacteria to now be able to metabolize citrate, it should have been a much more widespread thing. So it's actually arguing for evolution in both directions. The fact that it can happen at all with mutations that we know happen randomly and that are consistent with this large number of mutations that are happening across all of these trillions of bacterial cells, that it happens at all means that evolution can indeed proceed via mutation and non-random selection. And it also means that, in fact, because it's not universal, there is unlikely to be any intentionality behind it.
And he makes a good point there, Ahmed. We addressed that. So let me make sure we have the question on the table to be specific here. If it occurred twice in all these trillion things, where's the intentionality come from?
Okay. Where is the intentionality? It occurs, number 1, it occurs twice out of 5, if I remember correctly. So I think
there was 10. I think there was 10 baskets, if I remember correctly, but irrelevant.
Where the suspicion of intentionality is that you are not looking at a mutation that does the whole thing. Number 1, it was not a mutation. The citrate transport gene was there already in the genome. It just moved from an inactive promoter to the active promoter. So
that's mutation, though, would it not be?
Yeah, transposition is a mutation.
This is like a transposition, but it's not like a base pair has changed. And number 2, that only the transport was enabled, so the citrate can come in, but the whole rest of the metabolic cycle of citrate was already programmed. So once citrate was in, it was being metabolized.
Okay,
so it is there, the machinery is there to metabolize citrate like it's there to metabolize.
Let me see if I understand the argument. So I think your argument right now is that perhaps these mutations, because they are mutations, transposition, duplications, these are mutations, that's fine and dandy, but because it didn't produce any particularly novel function, it simply reactivated a function that we suspect was ancestrally present in E. coli, but that no longer is, it's not enough. It doesn't do enough. Is that a reasonable assessment of your argument?
By the way, I don't mind if we call them mutation. My issue is if they're random or not. So we can call anything a mutation, I don't mind. Transposition, deletion, addition, insertion, I don't mind. Call it whatever you want. It is not random. This is the issue. Randomness is not direct to show. This
What
is
makes
the
it
issue.
not random? How do we know that it's not random? Because we know that transpositions occur randomly, we know duplications occur randomly, and that's what happened in this experiment.
We assert that they occur randomly. Maybe this is an argument from incredulity, because
Well, we don't have a way
when you have bacteria, and bacteria tend to be very efficient, having all the machinery for citrate, only the transport is missing, and then it gets turned on in the right place. Invoking randomness here looks absurd. You have to agree with me on that.
No, I don't.
You will not have a power station working on diesel, and it has all the equipment for working on natural gas for no reason. And then when diesel is out, somebody opens a valve and natural gas is coming in, and we call this a random event. This is exactly what happened.
Okay, well, here's a few things. One, factories being run by intelligent humans are not a very good analogy for a bacterium. I mean, there are ways in which there are similarities, but just the fact that the humans running around the factory know what they're doing makes this a bit of a disanalogy here. But the other thing is, if it were a large group of the bacteria that had this mutation happen, then I would agree that would be somewhat suspicious that this large group of bacteria all had the exact same mutation. But that's not what happened. Even in the flasks which develop citrate metabolism, what would happen there is not that the entire flask would have this overnight. 1 or 2 bacteria in the flask would start out this way. But because of the fact that after a short amount of time, essentially all the glucose was gone and all that was left was citrate, they would then come to predominate the population in the flask, which is in fact what was actually observed. Because in the experiment, after the glucose supply was running low, the flasks, which started out cloudy because of the bacterial explosion in population after only a few hours, would then clear out again because most of the bacteria had died. But in these citrate metabolizing flasks, the cloudiness persisted for a very long time because, in fact, they were eating the substrate, which was not previously digestible. So you don't actually need a large number of bacteria to have these mutations. In fact, if it happens in 2 flasks, we need a maximum of 3 bacteria total. 1 to have the priming mutation, and 1 in each flask to have the final mutation that actually allows citrate transport. There is no reason to think that this isn't random, simply because we have billions of bacteria undergoing mutations, and we can check other bacteria and see that they're having similar but not useful mutations. In order to say that this couldn't be random, you have to show why, and your incredulity is insufficient.
The why is that all the billions of the bacteria have all the machinery to complete the metabolic cycle. So to say that complete a cycle with an already existing part of the genome is a random event
what if we had an organism that gained an entirely new protein that it didn't have before and that gave it a new function? Would that be of note to you?
Okay, so let me be very clear here. The matter of the phenomena of mutation is something that happens. Okay,
right.
It is very rare that this thing would generate something of benefit, but it might happen. Now the question is that when this thing that comes that is of benefit, this new protein that you talk about, and then this new protein comes about, if it's of benefit, it means that the system around it is ready to receive it. There is a function waiting for it to happen. Now this makes it way more difficult. Okay, things that are way more difficult can still happen, but can this thing that is extremely now difficult to happen explain the origin of species? This is the question. So I will give you a final example. So sometimes something like sickle cell disease becomes beneficial. Okay, it is a disease, it is a deformity in hemoglobin, it causes red blood cells to become sickled, it causes people to have plenty of problems and plenty of pain and a horrible life, but it also causes the malaria parasite not to be able to get into the red blood cell. So under a malaria situation, this is beneficial for people so as not to die of malaria.
Yeah.
But the issue here is because I'm referring here when you were talking about being at the peak of efficiency or at optimum fitness or not. Mutations, we always see mutations breaking things. Even bacteria resistance is we saw them very frequently. Mutations are breaking things, causing deformity, diseases, etc.
It's
They
simply
can't.
untrue to say always. And there's a
Actually,
big
I have looked and looked.
okay, well, let me help
yeah,
you. So, do
yeah.
you know what Antarctic ice fish are?
I just want to complete this sentence. I think you are going to invoke the matter of antifreeze proteins, etc.
I might,
But
because
now, yeah. So now what I'm saying here is that the vast majority, even population genetics asserts this, the vast majority of mutations will be deleterious.
no,
Some
neutral.
will be
The vast majority
some will be neutral if you acknowledge the matter of having a good part of the DNA doing nothing, and then a change to a coding sequence will then do nothing.
we don't have to acknowledge that. We know for a fact that most of the mutations that occur in populations have no measurable effect on phenotype because we can measure the mutations and we can check to see if there is an effect on phenotype. We know for as much confidence as science can ever give anything, that the majority of mutations that occur do not affect the phenotype of the resulting organism.
Okay, we got to start wrapping it up here, guys. We do have only about 5 minutes. What I'm going to do is this.
Do we have Q&A left, or
what's that?
are we still doing is there more Q&A or no?
A couple of minutes of.
Yeah, no, I'll give you plenty of time. I got a super chat I got to get to. Okay, so let me get that out of the way. Inkboy, $5. I would, as a question in the super chat, but Dapper Dino is giving the question, so no question in the super chat. I'll make a comment that even creation.com recognizes what's called the Delta CCR5 Delta 32 as a beneficial mutation, so not all of them are deleterious. Go ahead. If you want to take any more Q&A, I know you're going to have an after show on your channel, Dapper. The link is in the video description, and people will find it over there. Do you guys want to take a few questions? I don't mind going maybe 10 or 15 more minutes. I wanted to kind of have a hard thing at 2 hours, but we can go over if you guys are up to you guys.
I'm okay to take a few questions if people have any.
Okay, so Ahmed, did you want to do that? Go ahead and go. By the way, finish anything you want to finish up. I want to make sure you have plenty of time, and then we'll go from there.
So just to wrap up and make sure that, because we've gone through plenty of discussion, what I'm saying here is this. We have a theory that claims to explain the origin of life. It means it is putting itself in a position to explain how life has evolved from a last universal common ancestor until the human being. And what we see on the ground is that in all or the majority of the major forks on the road from Luca to human being, other mechanisms other than mutation are invoked. We do not see in operation that mutations can produce these kinds of effects. We see in operation that mutations can be sometimes apparently beneficial, but in the core of what they are doing, they are essentially deleterious, like the case of sickle cell. Some mutations might be beneficial, but they are point mutations. Maybe we later find out that the antifreeze protein was not really a mutation, it is something else. But even if it is few nucleotides that changes from this kind of protein to that kind of protein, the question now is, does it make sense that, is it even mathematically possible that a random process, a process that is based on errors in reproduction, would line up so many mutations all in a row, so that you start from Luca over, or actually since the Cambrian explosion or a little behind it, you start from unicellular organisms, and then within a few million years you have all this kind of diversity that eventually leads to the human being after a while. I think that this is a stretch. I think that human knowledge, as we stand today, is so behind. The more we know that we do not know. To the extent that continuing to invoke randomness as the driver of life, as the innovator of life, and natural selection as the filter that directs this to the right place, is so much of an offshoot. This theory needs serious revision. The longer we continue looking at randomness as the source of life, the longer we are missing opportunities to find real mechanisms and to find real progress and to give real benefit to the human race. This is my position. I'm not denying anything. I'm just denying that disorder is what generates progress.
All right, I got about three questions here. One of them is pretty straightforward. Do you agree natural selection is not random?
Natural selection is not random as far as the biology of the organism itself is defending itself. Natural selection on its own is not a thing, but it is not random as far as the status quo. But when we are looking at progress, then you are looking at moving from point A to point B. Natural selection has no point of view whatsoever to where you are going. So it cannot take you there.
It's not goal driven.
I'm sorry?
You're saying it's not goal driven.
It's not goal driven.
Okay.
There is no objective function for natural selection.
Not
like a genetic algorithm where you are making trial and error for a certain goal.
Okay.
It might as well take you down the road like it does. It favors cheaters in a colony. It will favor people getting sickle cell disease if malaria is prevalent. But what happens when malaria goes away? These people will suffer.
All right.
And this is not the kind of mechanism that will create the human being.
Eating Point wants to know for 2 pounds.
Whatever that is.
Do you accept, is guided evolution your position? Guided evolution. And that kind of goes into one of my questions I have as well, but do you think it's guided?
So guided evolution has a theological aspect, so I will just say very few comments here. Now, some people would go say guided evolution, and when they use the word evolution, they are invoking the mechanisms that are coming behind evolution. And the primary mechanism in macroevolution is randomness. So what they're essentially saying is that God, who will be guiding, because nobody else is guiding, unless even aliens or something. So if the person who is guiding this, who is God, is using randomness, what I am saying here, that if you are coming to this from a theological standpoint, you are essentially asserting that God is intentionally obscuring himself, so that when you do your research, you will find that the hardcore of the process of progress of life is randomness. And the definition of randomness is that any future event has no relation with the present or the past events. This is what randomness is. So you cannot penetrate this shell of randomness. It means that God is completely obscuring himself when it comes to biological progression of life. This is not consistent with any religion that I know about. It's definitely not consistent with my religion. In my religion, in Islam, God gives a specific instruction to Muslims to walk the earth and look at the origin of creation. There is actually a verse specifically asserting this. And if this verse is there saying specifically this, it means that when we look at how creation has occurred de novo and how it has progressed, we will find signs of intervention, of intelligent creation. We will not find randomness. So the people who are believing in guided evolution, I think they will have to either redefine randomness or redefine guidance. Guided evolution is not compliant with the theory of evolution, which invokes randomness. This is my position.
I think you might have. I think we might be a disagreement on what is meant by random, because a lot of people would probably think random, and I'm one of those people. Random is stochastic. Random means it could be a deterministic thing, but we are unable to be aware of all the information. For example, let me ask you this, because it's similar to what you were talking about before. But let's say that I flip a coin. Once that coin leaves my hand, it's a deterministic process. It's guided by the laws of physics, okay? But I'm unaware of what the outcome is going to be. I don't know. I have no information about the system, whether it's going to be heads or tail. It is a perfectly random system, stochastically to be, because I don't know the outcome. But it is deterministic, right? So I think it's fair to ask, how would we recognize a mutation that has some intentionality behind it, by which it was somehow made to be the case because of some, I would say, a mind, because in order to have intentionality, you have to have some kind of mind to do it, as opposed to just flipping a coin and whatever the results is based upon the laws of physics, but we're unaware of it. How would we recognize the difference between an intentional whatever, making that coin heads or making that coin's tail, as opposed to a stochastic random process, which is just missing information about the system, the outcome of it, and then apply that to like the Lenski experiment? How would we know that is intentional rather than just a stochastic process where we're just not able to have the information to determine it, even though it is a deterministic process?
Your point is clear. It's very clear. But this is exactly my point. It is because what we call random are essentially processes that are, by their physical nature, completely unpredictable. So why would God tell us, look at the origin of creation and you will find me? If those processes are random, or random to the extent that we cannot know how they happen, how can that be evidence for God? So what I'm saying here is that as far as we're concerned, those processes are random in the definition of randomness that can be modeled by a Markov chain, for example. And this means that no matter how you want to find intent behind it, you cannot. This is the whole issue. It is random because you cannot find intent behind it, because there is no causal relation between past, present, and future in a random process. So this is the whole point. So people who, for example, in my religion, people who invoke something called occasionalism. And what they're saying is that, yes, it looks random to us, but essentially God is managing it from behind the curtain. Well, if this is the case, he wouldn't point you to it. He wouldn't tell you, go there and look for me and you will find me, because he knows that he has created it in a way that you will only find randomness. So I do not believe in something called guided evolution. I believe that if God wants to guide the process of evolution of biological evolution, he will cause the right events to happen in the right time, like what happened in the Lenski experiment. He doesn't need to cause a trillion gazillion creatures to die due to mutational diseases to obscure himself. God is not in this universe to obscure himself. He is in this universe to be found. This is my theological position, and guided evolution is a theological standpoint. It's nothing more than that.
All right. So we're gonna, like I said, we're gonna wrap this up a little bit. The question was asked earlier about a paper. If you guys had to each suggest one paper, what would you suggest? And there's no way in hell I'm going to get into Photo's argument right now, Icarus. We've all been there, done that. I actually think Photo's argument is really interesting. Actually, I think that people poo-poo it away too quickly, and they shouldn't, because Photo's argument is very good about natural selection. But that's a whole different video there, buddy. I think you would agree on that, Ahmed, obviously. So if you had to recommend a paper, Ahmed, what would you recommend? Then I'm asked Dapper the same question. Give you guys about 5 minutes each to kind of do a recap, and then you guys can head over to Dapper, because I mean, this is a never-ending topic. There's so much to talk about. You guys have put so much on the table, and I first of all, I want to say I applaud both your guys' ability to communicate on this topic very, very eloquently. I miss these types of conversations. They were amazing that we used to have the Non Sequitur show, and you two both typify what we would have on the Non Sequitur show to have these kind of discussions. So I want to thank both of you guys for that. But go ahead and tell us what paper you would recommend to people, Ahmed, and Dapper could do as well.
Well, I actually would recommend the exact papers that we were talking about. I would recommend people to read the De Novo Origins of Multicellularity paper, to read the paper where Lenski did his work on E. coli, and find out for themselves if there were any real transition from single cellularity to multicellular organisms. If there are really a random event that has driven E. coli to metabolize citrate. And I think people reading about, we don't need to read to notice that, for example, human language, human intellect, and human free will are nothing like any other animal. So where is the gradient that is predicted by the theory? And putting those three together, one that looks at the complexity of the eukaryote, one that looks at, which was, you can look at videos like what we have seen, people who have modeled the molecular machines. Those are really amazing. And then the papers, the couple of papers that I've talked about, and then the very observation that there is no gradient whatsoever between humans and anything other than humans. Nobody talks like us. Nobody makes this eloquent conversation like us that Dapper did. I will not say that mine is necessarily eloquent. Nobody thinks like this. Nobody has this scrutiny and criticism. Nobody goes out of his way and rebels and says today it's this kind of world, tomorrow it's capitalism, the day after it's the other thing and the other one. No, we make our own rules. We have free will. Where is this in the rest of the animal kingdom or the rest of the creatures living around us? We are not the product of a continuum of a smooth gradient of mutations. There are very big jumps in evolutionary biology. I will not call it evolutionary biology. There is a design, I would say that looking at embryology, where a single cell, a sperm and an egg, will merge together into a single cell, and it will unfold to make a human. This is how creation is. No randomness there. The matter that it unfolds over 3 billion years or 500 million years or 9 months is not material. The thing is, there is a designer that made this thing who is able to make it unfold intentionally through design, through programming, that can unfold over 9 months and can unfold also over a few billion years. This is my position, and this will be very obvious if anyone looks at the material of the evolutionary biologists themselves, armed with some mathematics and some logic, putting prejudice aside. I think it is not difficult. It's the same evidence that everybody's looking at. It's an epistemic matter. It's a matter of back the worldview. It's a matter of epistemology, what interpretation you reach. And just commenting on what you said, I would like to thank Dapper. I think this is a respectable person to have a conversation with. This conversation is what I hoped it to be. And I'm glad that you moderated this, Steve. It was really fun.
Thank you. I appreciate it. And likewise. We're going to take that as your kind of your summation, I guess. I think that was well elocuted. So, Dapper, I'm going to give you the same amount of time to kind of wrap it up. And you can take it over to your channel and continue on. Because like I said, there's a million different things you guys could talk about. And I only got two hours plus, not 7. I am going to be on Drunken Peasants tonight to throw my own thing out there. People want to watch at 6 o'clock. I'll be on with Ben tonight. That's going to be fun. Don't know what the topic is. I never do, and they never tell me, which is scary. I don't like going into things ill-prepared, but, you know, it's still kind of fun. But, Dapper, go ahead and wrap it up.
All right, so in terms of what I would recommend for reading, in terms of papers, I don't have a specific one, but I would encourage people to check out some of the papers about the topics that we discussed. So for instance, read papers coming out of the long-term Lenski E. coli experiment, read papers on the novel Endosymbiosis in Pollinella, read papers on the emergence of de novo multicellularity in Saccharomyces cerevisiae, brewer's yeast. So check some of those out, because all of those will give you the actual hard numbers. But if you want a more lay level read that will show you the deep, deep connection between humans and non-human animals, I highly recommend Neil Shubin's Your Inner Fish. I recommend it to everyone. It will go a long way towards describing exactly how it is that various developmental genes result in wildly differing anatomies that are yet still based on the same genetic basis and the same basic structures. You can watch experiments where they gave stingrays fingers. You can watch experiments where they you can read about, things like giving alligator embryos feathers by just subtle tweaking of various developmental genes. It's fascinating. And I think if you read that carefully and take it on board, you probably not see developmental biology or evolution quite the same way anymore.
And I'm going to throw that in there. I happen to think You're in a Fish by Neil Shubin is one of the best books I read on phylogeny and these types of concepts. Brilliant book, well written, well explained. It doesn't matter you're a creationist, evolutionist. It doesn't matter. It explains things very well that anybody can understand, and it is one of my favorite books as well.
As far as a summary, basically, I think we primarily heard almost entirely incredulity from my debate partner here. Many of the things that he said we don't have examples of, we not only have examples of, we have examples of them both in the wild happening now, as well as in laboratories. I have yet to hear a good reason as to why we can say that mutations that are beneficial aren't random anymore, or why it is that random mutations cannot actually produce complex structures or produce beneficial mutations. There was an assertion of intentionality that I don't think was sufficiently well backed up, and that was in fact contradicted by the actual preponderance of the evidence. So basically, I don't think that my opening statement has really been assailed. We know that descent happens. We know what happens with modification that is caused by mutation. And we know natural selection filters out some of these modifications, and that the ones that aren't filtered out are the basis upon which further descent with modification occurs. I think that this has been a fairly solid and easy well, not necessarily easy, but a fairly solid win for the affirmative position on the resolution.
Well, let's see what the audience has to say in the comments section. I'll keep you guys both posted. Both of you are in the Great Debate community on Facebook, and I'm sure the conversation will go over there. And Ahmed, I would love to have you back to talk about some of the epistemological concepts that you had brought up, because that's kind of more my jam than the biology. Although I love biology, I'm obviously familiar with this to an intro level degree, because I've had so many of these discussions. Dapper's the guy I go to when I need to find out something. But I do like epistemology a lot. So I'm happy to have you back for that. If you guys want to join the Great Debate community on Facebook group, by all means, pop in there. We have great discussions. It is unfortunately a relic from the Great Debate community on G+. But hey, a lot of my audience has been around for 7-8 years, and they're well aware of all that. So with that, guys, I want to thank you both very much for coming in. Again, I am very impressed with both of your guys' knowledge levels, your elocution, your argumentation, and the fact that this didn't have to turn into a dumpster fire, and it sure didn't, and it was productive, and you guys were great. Let the audience decide, of course, who they think has a little bit better argument. That's how it works. But I love the fact that we can all get together, we can have these discussions, and people know by now I moderate the way I do Non Sequitur show when I have these types of discussions. It's not just, letting two people speak. It is an interaction between the host, the mods, and the people listening. It is a symbiotic thing of endosymbiosis between a bunch of symbiotes that we all are getting together and for the benefit of the species. And I'll leave you guys with that. And good night.
Thank you, Steve.
Good night.
Awesome. Good night.
Good night.